So the skin cells are very, very long and thin in a dish. After we add the different genes to them, they begin to change morphology. They become a little bit looser, they become a little bit rounder, but they're not really real stem cells yet. However, after about 5 weeks after we begin to treat them, what we have appear is something that has a very characteristic morphology. And what we have here is cells that are very round, they're very tightly packed, and there's a very distinct border that surrounds these colonies, and we can see they're very different from the initial skin cells that we started from. This gives us an idea that we probably have stem cells. But we need to be sure, are these true stem cells with all the capacities they should have? So what we need to check is, are they pluripotent, so the capacity to become anything they want, firstly at the DNA level. And we know now there are many genes that characterize stem cells. So what we do is we take different genes and we look if we can see these genes are actually turned on in these cells. So here, it's different techniques we use in the laboratory. But if you see a little black band here, it means the DNA of that gene is turned on. The gene is expressed. So we can see here in our iPS cells, we have these 4 genes that are turned on, whereas just before in the initial cells, the fibroblasts, the skin cells, these genes aren't turned on. So something has changed. That's at the DNA level. And then there's the protein level, because DNA, genes give proteins. So do they express the proteins that indicate they are true stem cells? So what we have different staining methods. We can stain our colonies, and if they become pink, that means they're pluripotent. Here again, it's another marker. If they have a red signal, like in blue is the nucleus, as Jean mentioned yesterday, that contains the DNA. In red is the expression of a specific protein, a pluripotent protein. So this indicated to us that we had iPS cells from a patient that were real stem cells. But the next question was, were they really still CHM cells? Do they still have the mutation that the patient had? So before I go into this, I just revise a little bit the DNA so it's easier to follow. As you may know, DNA with the genes are made up of what's called exons, these little boxes here. The CHM gene has 15 exons. However, in between exons, we have what's called introns. So it's DNA sequence that doesn't mean anything. The cell doesn't understand this. So to be able to read a gene, a cell needs to get rid of these introns, and this is called splicing. And it makes an intermediate molecule called an RNA. So here the 15 exons are put together, and suddenly the machinery of the cell can read this like a sentence. It makes sense to it. And then it makes its protein.